Okay, I guess I'll just start. So hello, everyone. My name is Julia Haag. I'm a PhD student at the Heidelberg Institute of Theoretical Studies. And in my presentation, I want to talk about simulations of sequence evolution, and especially our study on the realism of simulations of sequence evolution. And before we start, I want to briefly motivate why we even started studying the realism of sequence evolution simulations. And to do so, let's talk about how AI works. So basically, we feed a bunch of input data into a trained AI, and then magic happens. Or as we would maybe like it in phylogenetics, we feed in multiple sequence alignment into a trained AI, and then we get the hopefully true phylogeny. What would we need to do to train an AI that can do this? Well, we would need a lot of training data. So we would need a lot of MSAs and the ground truth phylogenies. That is, of course, a bit of a problem because for empirical data, we don't have the true phylogenies. The whole point of training an AI is to obtain these phylogenies. So can we use simulated data? For simulated data, we do have the multiple sequence alignment and the respective phylogeny. But are simulations actually good enough in representing empirical data? So are simulations realistic enough to train an AI? Because if we're training an AI with data that is not like empirical data, that doesn't seem to make a lot of sense. So this was actually the question, the research question we had: how realistic are simulations of sequence evolution? And this presentation, I want to show you how we assessed the sequence simulation realism and what we found out. The very basic and high-level concept of our assessment strategy was to train classifiers that get an MSA as input and then predict whether the multiple sequence alignment is simulated or empirical. In a bit more detail, so what we did is we selected a model of evolution that we want to assess the realism for. We also select a number of empirical multiple sequence alignments that we can simulate along, but that we can also compare against. So we need to train our classifiers with simulated and empirical data. So we, of course, need to also simulate the alignment. So we have the empirical data, the model of evolution, and we simulate a new collection of multiple sequence alignments. Then we train classifiers that learn to discriminate between the empirical and simulated alignments. And using these classifiers, we then evaluate the realism of the models of evolution. And basically, the idea is the easier it was for the classifiers to discriminate between empirical and simulated alignments, the less realistic the model of evolution is, the less realistic the simulations were. How did we actually simulate the data? So for data simulation, we simulated a total of 15 data collections. So 15 collections of simulated multiple sequence alignments. We simulated 7 data collections for DNA data and 8 for protein data. We used increasingly complex models of evolution. What I mean with complexity here is, for example, the number of free parameters that the models have, or the computational methods that were used to derive the respective model of evolution. And for DNA data, we started as the most basic model of evolution with the Tukes-Cantor model. It has equal base frequencies and substitution rates, and we increased the complexity up to the general time reversible model with 4 rates for gamma site heterogeneity and an additional free parameter modeling invariant sites. For protein data, we also started with the most basic model with equal base frequencies and substitution rates, the Poisson model, and we increased the complexity up to the universal distribution mixture model with 256 profiles and a continuous gamma distribution to model site heterogeneity. Now, as I said, we also need empirical data to compare against, but also to simulate along. So we wanted to have as realist or as representative data as possible. So for DNA, for our DNA studies, we used MSAs we obtained from Treebase. Treebase is a data set of published empirical alignments, and there are no limits to the sequences that can be published there. So it's a very heterogeneous set of alignments that basically are a good representation of the data that is typically analyzed in phylogenetics. And for protein data, we used MSAs we obtained from the whole genome database. This is a database of sequences from bacteria, archaea, and eukarya. So we also have a very diverse set of multiple sequence alignments here. Now, to actually simulate alignments, we first inferred a phylogeny on the empirical MSAs. For each empirical MSA, one phylogeny. And the idea here was to prevent the problem of also simulating realistic phylogenies. And we also based other simulation parameters on the empirical alignments, for example, the best substitution model estimates or indel parameters. And we simulated the alignments using the AliSim simulation tool. So we really used the state-of-the-art simulation tool to get the alignments as realistic as possible. We did a lot of simulations without indels, and the idea here was that when simulating data without indels, we can assess the realism of the models of evolution per se. But we also, of course, simulated alignments with indels, as this is the data that is typically analyzed. And to simulate indels, we use two different approaches. The first one we called the mimic approach, that basically, with this, we basically simulated alignments the same way as we did without indels, and we then superimposed the gap patterns of the original alignment, of the original empirical alignment, onto the simulated alignment. And with the second approach, the Sparta approach, we estimated empirical indel parameters using the Sparta ABC tool, and we then drew indel parameters from the joint empirical indel parameter distribution to actually simulate the indel events. So as I said, we simulated a total of 15 data collections. We have 5 gapless data collections for DNA data. We have 2 data collections with indels for DNA data. For protein data, we have a total of 8 data collections, 7 of which are gapless and 1 contains indel events. Okay, let's talk about our classifiers. What classifiers did we actually train to assess the realism of simulations? And we used 2 distinct approaches. We used the classical machine learning approach, gradient boosted trees, and we used the deep learning approach, convolutional neural networks. And the idea of using 2 distinct classifiers was that we can leverage, or we can kind of have the best of both worlds. We can have the explainability of classical machine learning approaches with manual feature creation, but we can also have probably a better performance using deep learning methods. And also CNNs require only minimal data pre-processing, so we don't need the handcrafted feature as we need for the gradient boosted trees. We trained 1 classifier each for each of the simulated data collections independently. So this is a very important part. We trained all classifiers independent of each other. So we, in the end, obtain 15 trained gradient boosted trees and 15 trained convolutional neural networks. To train the classifiers, we combined the simulated data collection for the model of evolution that we want to assess the realism for with the respective empirical data collection. And we used a 10-fold cross-validation procedure for training, and we report the balanced accuracy as average over all 10 folds as our final realism indicator. We use the balanced accuracy because this is a metric that is invariant to class imbalances in the data. Now let's talk about in a bit more detail about our classifiers. So first, let's talk about the gradient boosted trees. As I said, this is a classical machine learning approach, so we need to handcraft features. And we basically used 3 groups of features. We used a total of 23 features, not going to go into detail on all of them. I just want to give you a high-level overview so you can have an idea of what kind of features we're looking at our MSAs. And the first one we call the branch length features. And these features are based on a single RexMLNG tree inference for the MSA. And then we extract branch length information, so branch length summary statistics from this phylogeny. The second group is based on our difficulty prediction tool, Pythia. Basically, we use, for example, the predicted difficulty. So how conclusive do we expect a tree inference for this multiple sequence alignment to be? But we also use basic attributes of the multiple sequence alignment, like the sites per taxa ratio or the proportion of invariant sites. And the final group that we call the randomness features, is based on a recurrent simplification of sequence assimilations. That is the assumption that evolution occurs at uniform distributed sites in the MSA. So basically, we expect simulated MSAs to be more uniform in terms of, for example, substitution counts than empirical MSAs. And we're quantifying this using a random sequence testing. And for example, one metric that comes out of this random sequence testing is the serial correlation coefficient that basically states how much the substitution count at one site depends on the previous site. Now, for the convolutional neural network, I don't want to go into too much detail about the architecture. It's basically a very simple CNN architecture. We don't have too many layers and not too many trainable parameters. There are just a few things I want to highlight here. So first of all, we encode the multiple sequence alignment in a numeric data representation. And for this numeric data representation, we compute the frequency of each character at each site. So what we get for DNA alignments are 5 vectors corresponding to the 4 nucleobases and 1 vector for gaps. And for protein data, we get 21 vectors, so 20 for the amino acids and 1 for the gaps. For DNA data, we do an additional embedding step that we don't do for protein data. And for this embedding step, we're using a convolutional kernel of size 3. And the idea here is to capture potential codon structure in the DNA data. Okay, so what is actually the performance of our classifier? So how realistic are the simulations and the models of evolution? And what we observe is a remarkable overall accuracy. We don't need to go into detail on all values here. It's just for the sake of completeness here. But what we see is that except for one model, all of our classifiers achieve a balanced accuracy higher or equal to 0.89. And with 1 being the best possible score, this is really a remarkable accuracy. So we conclude that in general, the simulations we did and the models of evolution are not realistic. So even for the most complex models, so as I mentioned before, we had the hypothesis that more complex models of evolution might result in a higher simulation realism, but we don't really see this, as all of our classifiers are actually very accurate, especially the CNN classifier was easily able to distinguish between empirical and simulated data. So why is that the case? Why do simulations seem to be so unrealistic? Why is it so easy for our classifiers to distinguish empirical and simulated data? And to find out, we can use the gradient boosted trees and the feature importance. This feature importance basically states how much a feature contributes to increased performance during training. And what we observe across all of our gradient boosted trees, across all data collections, is that the randomness metrics are very important. And one super important metric is the serial correlation coefficient that I mentioned before. So how much do the substitution counts at one site rely on or are dependent on the previous site? And we can nicely visualize this using 2 exemplary MSAs. We have an empirical MSA on the right side and a simulated MSA on the left side. And basically, the brighter the color, the higher the substitution count at a specific site is. So it's clearly visible that the simulated data is more random in terms of substitution counts than the empirical data. Now, these randomness metrics, these randomness features mostly quantify the randomness across sites. We've also seen other metrics that measure randomness within sites among the most important features. And we will talk about this randomness within sites in a few seconds when I talk about the CNN prediction performance. But we also observed other interesting things with empirical data. For example, for empirical data, the proportion of invariant sites tends to be higher than for simulated data. And also branch lengths in phylogenies inferred for empirical data tend to be longer than for phylogenies inferred on simulated data. So let's also talk a bit about the CNN feature importance. And actually, it's not as easy to obtain a feature importance for convolutional neural networks because it's a deep learning method, and we've heard it before, it's a black box. But we had the hypothesis that the convolutional neural network has some kind of global feature that is indicative of the simulations or of an MSA being simulated or empirical. And our hypothesis was that this is the alignment-wise amino acid or nucleotide frequency. So what we did to test this hypothesis is that we trained a logistic regression classifier. So again, a very basic classical machine learning classifier using only this feature. So using only the alignment-wise amino acid or nucleotide frequency. Now for DNA data, this did not really work. So we never got a classifier that achieved better performance than random guessing. But interestingly enough, for protein data, this worked incredibly well. So even with this very basic logistic regression classifier using only the alignment-wise site compositions, we could distinguish between empirical and simulated MSAs with high accuracy. So this MSA composition of sites seems to be highly informative for simulations versus empirical data. So let's summarize what I've shown you and discuss this a little bit. So we have seen that we have an overall remarkable accuracy of our classifiers. So we conclude that we have in general a low simulation realism across all models of evolution, no matter the complexity of the evolution, of the model of evolution. I want to mention again that we put a lot of effort into simulating as realistic as possible. We used representative empirical data sets, and we still got the low simulation realism. We used two distinct approaches. We used CNNs and gradient boosted trees. They both use different sets of characteristics. So with gradient boosted trees, we have these global features, the handcrafted features, and with CNNs, we have the site composition as a feature. And when we look at the feature importance, we really see that the assumption of randomness or the assumption of uniformity of evolution in sequence simulations is a big contributing factor to unrealistic simulations. So what does this mean? Where do we go from here? Do we just stop doing phylogenetics altogether, or is there any other way? What can we do? So for one, maybe we can start thinking about developing better models of evolution. Or maybe we can start developing simulators that don't rely on an explicit model of evolution. So maybe we can use deep learning approaches like generative approaches for this, where we don't have to specify an explicit model. Our approach, our method could be used as a testing framework for future studies on new models or new simulators. Because if you train a new classifier and the classifier on your new sequence simulations has a high accuracy, that indicates a low simulation realism. But I also want to mention that a low accuracy of a classifier does not necessarily indicate simulation realism. It could also be due to other confounding factors. For example, problems optimizing parameters of the deep learning methods, or maybe inappropriate data representation, or simply missing features. Now, as a final note, maybe all this is not that new. So we, of course, wouldn't expect models of evolution, explicit models of evolution, to perfectly capture the complexity of evolution. But at least to us, the unrealism of data, so how easy it was for our classifiers to distinguish between empirical and simulated data, that was surprising to us, and maybe it was a bit surprising to you as well. Thank you.